Hi, everyone. My name is Tejas, and I worked with Dr. Kristenu Bandapadhyay on our project titled The Bioconjugation of Gold Nanoparticles. So I first want to talk a little bit about the background around our project. So in the past recent years, there has been a large increase in the research into gold, silver, and palladium metal nanoparticles for usage as vehicles for drug delivery to different cells throughout the body and in the brain to allow for helping with protein synthesis and ER stress. So proteins are synthesized at the endoplasmic reticulum in our cells and are used for a variety of functions, from our hair to our skin to our eyes, and are necessary for our body to carry out normal homeostasis. However, disturbances in calcium regulation, glucose deprivation, and viral infections can cause certain proteins to misfold and lose their function and lead to a condition known as ER stress due through oxidative conditions and reactive oxygen species. When cells detect ER stress, they trigger the unfolded protein response, which can trigger the initiation of cell death and apoptosis, and is actually one of the leading causes of Alzheimer's and Parkinson's disease, as it leads to neuronal cell death in the brain and eventual loss of function. However, ER stress can be combated using certain drugs, such as 4-phenylbutyric acid, which is shown in the image below here, which acts as a selective inhibitor of protein misfolding and can actually work as a chaperone to refold proteins and prevent apoptosis and cell death. A little bit more on our purpose. So how this works is that the blood-brain barrier is composed of a series of tight junctions between epithelial cells that comprise our blood vessels in our brain and actually inhibit the passive and active transport of compounds such as 4-PBA. And it was one of the reasons why we sought to begin this project in the first place. In the image here in the center from USC, I have an image of the tight junctions of endothelial cells that actually line our blood vessels, so you can get a better image of what I'm talking about. So our project sought to make 20 nanometer gold nanoparticles to use as a drug delivery vehicle for 4-PBA to coat the surface of the nanoparticles. So give me just 1 second here. Sorry, there we go. I just saw the background noise. We sought to make 20 nanometer gold nanoparticles, as I said, for usage as a drug delivery vehicle for 4-PBA, which we're hoping that can eventually be used as a treatment to suppress the unfolded protein response to prevent and alleviate neurodegenerative diseases such as Alzheimer's and Parkinson's in the future. So our procedure here, I have a diagram on the right and a little bit of it written out on the left. So the first step was actually to synthesize the 20 nanometer gold nanoparticles, which can be seen in part 1 right here. The second step was to form a monolayer surrounding our gold nanoparticles, which would then be used to conjugate our gold nanoparticles to the drug. The final step, number 3 here, was to use 2 compounds and form a cross-linking reaction in which our 4-PBA compound would actually bind the surface of the nanoparticles in a stable manner. So this slide talks a little bit of our synthesis of the gold nanoparticles. So briefly, we made a solution of a gold salt and heated that up to 100 degrees Celsius. While stirring at 400 RPM, we injected a solution of trisodium citrate. Upon that injection, you can see that from time 0 in the image on the top right here, all the way to 120 seconds, there was a large color change from basically yellow to gold to black to red. After 120 seconds, the color of the solution doesn't change anymore. The image on the bottom right here is a size distribution of our gold nanoparticles. You can see that the majority of our nanoparticles do fall between 21 and 23 nanometers, which is expected for the procedure and what we were aiming for. Here, I want to talk a little bit about our data and results of actually doing the conjugation. So in this box, which is different colors of green here, you can see that our size increases from going from the gold nanoparticles to our monolayer to the drug cap nanoparticles, which is expected because we are increasing our size through adding more molecules. The zeta potential, which is a measure of the stability of our nanoparticles, does decrease from -43 all the way to -15, which is also expected due to the addition of such large molecules surrounding nanoparticles. We see this in the fact that stable regular nanoparticles with a zeta of -43 can last from weeks up to months in storage. But once we actually conjugate them with the 4-phenylbutyric acid, they only last for about a week and have to be used by then. The UV-Vis at the bottom here, which is also shown in the graph on the right, is another way of how we confirmed our increase in size of the gold nanoparticles through each step of the conjugation. And you can see it goes from 522 to 25 to 26. And small changes in UV are indicative of large changes in size. So we're not expecting to see, something from 520 up to 540 or anything like that. The two images in the center here, the one on the left is a transmission electron microscopy image of spherical gold nanoparticles that were taken and does show them being around 20 nanometers. The image on the right here is another microscope. It's an atomic force microscope, which we also used to allow for detection in a physical parameter to see the 20 nanometer size distribution. Finally, I want to just discuss a little bit on our future work here. So the image on the top here at the left is a fluorescent probe that we're planning to attach. This fluorescent probe will allow for easier detection through an in vitro cell assay use and an in vivo assay to detect where these gold nanoparticles are traveling and actually allow us to see their uptake in cells. Just underneath that, we have our protein activity assay, which will allow us to detect whether or not our drug after conjugation is still active. Normally, the drug without conjugation to nanoparticles does interact with human serum albumin and triggers a conformational change, which is what actually allows it to be carried throughout the bloodstream and delivered to cells. We're hoping that we can use the same protein, and through its detection using UV vis, we'll see changes in conformation that will allow us to confirm the activity of our drug. And here's just a list of my references that I used throughout this project. I hope you all enjoyed the presentation. And if you have any questions at the end, let me know. Thank you.